Good evening, everyone. Welcome to the next yet another session of our MBBT. So, as yesterday we were just discussing very basics from the ninth and 10th class about the structure of DNA and further its properties. Yeah. So we will continue with that. We will discuss today various types of DNA, and then we discuss about various type of DNA, RNA. So in that sense, our nucleosides, nucleic acids will be over. The first topic of the main thing will be over. Then we'll turn head to the translation and transcription. And what we will do, that we will have our session till 9 or 10 or by 9 o'clock, and the last 10 minutes we could switch to Google Meet, so we can discuss further our things that we wanted to discuss, and make any queries, anything more face to face after the lecture is over. Yeah. So this is the actually that system that I brought here is for everyone actually. That's I learned from Germany. So in Germany, when we were learning anything, the topic that we want to learn, what they do is they start everything with very basics. With very basics they start, and they gradually increase the hard topic to the advanced topics. So the same way I have brought this topic. So let's start with the basics. Then we go to the hard topics further. Okay. So I think most of the students have joined us, so we can start. So yesterday we were just discussing about the major groove, minor groove. We have discussed about the rise. We have discussed about the tilt. We have discussed about the length of one turn, and we have discussed there are around 10 mononucleotides per turn. And we have discussed there is a central axis, and the diameter is around 2 nanometers. So based on that, this is the basically the structure of a bDNA. This is typically found in our body most of the time, and the other DNAs, the A and Z, that are exceptional. Z is the most exceptional, A is still find out. So we're going to discuss the same thing here today. So there are three types of DNA, that is aDNA and bDNA. Whereas aDNA, it is a right-handed helix, even the bDNA is right-handed helix. aDNA has 11 base pair turns, whereas the bDNA has 10 base pairs per turn. Then aDNA is around 2.3 nanometer in diameter, and this is 2.09 in diameter. So we can say that this is more wider. This is 2.3, and this is only 2. Yeah, and so that's why the diameter is more. It's more broader. So we can say the type A DNA is more broader, whereas the B DNA is in the middle range, and also from the height point of view, it has 11 base pairs in one turn, and this one has only 10 base pairs. So there is a one base pair is lacking here. So that's why it's more taller. Your one turn will be more taller than this B DNA. Whereas in the Z DNA, it's opposite the hand. It's a left-handed helix, and it's called Z DNA because of the Z, like this structure it has Z type. And in the A case, we saw that there's 11 base pair turns, and in this case it will be 12 base pair per turn. So it's even bigger. It's even longer than the previous one, but the broadness, it will be the uh, thinnest. It will be not as thick as it was ADNA or BDNA. So this is the most thinnest. So that's the difference between your ZDNA, 1.8 nanometer. So let's discuss these points now, the main differences in this table, about the main points. So before we discuss main points, just want to discuss that we have discussed about the minor groove, major groove, yeah, and from the BDNA and the ADNA, the minor groove become more bigger, more thicker in the ADNA, and the major groove become more smaller here. So that's one difference. And we can see that the diameter is big, and here it's a bit small, yeah. And also the rise thing I have discussed, the distance between two nucleotides, it's known as the rise. And the tilt is actually the angle between two nucleotides. If one nucleotide is like this, the other one belongs to here, the angle between the two here will be known as your tilt. Yeah. So these are the main points we need to be taken care. So based on that, and also our ADNA and BDNA, they are both right-handed helix. In these structures we can see, and whereas it is opposite, it's a left-handed helix, the ZDNA. So this is Z type. This is more into Z type, this ZDNA, and this is a normal, and this is a bit broader. Sorry, bit thinner. This is broader. This is middle size. So here we can. So the ADNA is short, yeah, in the height, but it's broader. BDNA is longer but thinner, whereas the ZDNA, it's elongated and slim. Okay, so these are the main proportion of it. How it is short and broad? It's a rise per base pair. So within the two nucleotide distance is 2.3. Here is 3.32, and here is 3.8. Yeah, it's the widest. This is the widest, longest rise distance between two nucleotides. This is somehow in the middle. This is the smallest, 2.3. And this is the diameter, that how much is the diameter is. So this one is around 23. So that's why this is 25, and this is 18.4. So that's why the slimmest, this is thinner, and this is broader. And the height wise, it will be the shorter, this will be the longer, 3.32, and this will be the elongated. So these two features explains that the main difference between your A, B, and Z DNA. Then the helix rotation sense, that how we have discussed that how is the rotation is happening. As we have discussed, this is right-handed, and this is left-handed. Yeah. So how many base pair helix repeat? So this is one base pair per helix repeat. This is one, and in this case is two. And how many base per turn of helix? How many base pairs are present? So in this case, in one turn, that is major groove and minor groove. Your major groove plus minor groove. So in this it has 11 nucleotides, in this it is 10, in this is the highest 12. So that's why it's the elongated. This is longer, yeah, and this is shorter also. Then further base tilt perpendicular, the distance between two nucleotides, the angle between two nucleotides. So here is plus 19, and this is minus 1, 2, this is minus 9. Okay. So also the major groove proportion, this is extremely narrow but very deep. In the ADNA, this is wide and with intermediate depth, and this is flattened out on helix surface, whereas the minor groove proportion is very broad, and shallow. This is narrow with intermediate depth and is extremely narrow but very deep. So that's the major difference between your aDNA, bDNA, and zDNA. So this might be helpful for someone who has exam of molecular biology or preparing for GATE exam. So these questions come in very tricky way. But if you remember each part, what is, you can answer them very easily from this table only. Alternate forms of DNA. You can score 2 marks actually in the GATE exam with that. So that's the major differences of your A, B, and Z DNA. Further classification according to the eukaryotes and prokaryotes, these DNAs could be linear and circular. The linear DNA, the normal DNA, it's present in the eukaryotic cells. Okay. And these are the ones, and they are the one has both ends are free. Whereas in circular DNA, it's circular, so the ends are not free because in circular form. And this is mostly present in the nucleoid regions of prokaryote cells. So that's how the eukaryotic DNA looks like, and this is the prokaryotic. I will not go in details about them so much. So let's answer these questions now, what we have discussed so far. Write down these answers in the chat box, everyone. DNA, stands for D-ribo-acids. So answer this one. Answer maybe two together so we can finish them fast. So answer first and two first. So DNA stands for, and what is the shape of DNA? DNA stands for, what is the shape of DNA? And what is the shape of DNA? Yeah, helical is there, but one more point you are all missing actually, students. Double stranded helix, DS. Okay, good. Most of the students have answered that nicely. Now let's go to the next questions. Who established the structure of DNA? And adenine always pairs with. Who established the structure of DNA? And adenine always pairs with. Let's finish with the basics, then we can start. Uh, yeah, Watson Crick will be better because Rosalind Franklin, he just discovered the X-ray region, but the Watson and Crick were the real person who have discovered it. So with adenine, it's thymine. Yes, right. Yes, yes, students. Very nice. Everyone, give me the right answers. Yes, Watson and Crick together. Very nice, very nice. Beautiful. So let's go more advanced. The sides of DNA ladder are deoxyribose and the side ladders of DNA. And guanine always pairs with. Tell me the answer, students. The sides of the DNA ladder are deoxyribose and guanine always pairs with. The side chain I'm asking, bache, the side chain. So here some students are giving the wrong answers. Yeah, guanine pairs with cytosine, that's right. And sugar always make groups with the phosphate group, not with the nitrogenous group. I'm asking about the side ladder, not in the center. So it will be the phosphate group and cytosine. Very nice. Very nice. Extremely good. Extremely good. So next, what is the complementary sequence? Just tell me fast, quickly, students. AAT, GCA. So what will be the complementary sequence of this DNA? AAT, GCA. Just answer this question. AAT, GCA. AAT, GCA. Right, right. No, no, बच्चे cytosine not. TT, A, CGT. TTA, CGT. So two sides of DNA are held together by the bonds. The two sides of DNA are held together by the bonds. Second question, DNA is composed of repeating subunits of DNA is composed of repeating subunits of, and two sides of DNA they are held together by the two sides of DNA they are held together by hydrogen bond. Yes. Right, 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 right. Good. And and DNA is composed of repeating subunits, that is called as DNA is made up of repeating subunit, that is known as nucleotides. Right. Nucleotides will be the answer, bache, not nitrogen bases. Nucleotides. Good. All good. Everyone is giving the right answers. So next question is, what are the four bases that makes the rungs of DNA ladder? This is very simple. What are the four nitrogenous bases which makes the ladder of your DNA? Which makes your ATGC. Yes, that's it. So we are done with the very basics of molecular biology today, with the structure of DNA, various types of DNA, and have a little bit of Q&A here. Sangam Desai, very nice. You have written in different form, purines and pyrimidines like this. Very nice. Appreciate that. Um, so we will not make our time consumed in any other things. We will come. So in this case, the outcome is that you got to know what is structure and function of DNA. And now we can correlate this part of DNA with replication, transcription, and translation, which will be very helpful for further lectures for us. So these are references. I will discard this. Good. Very nice. So I have discussed these lectures already with you yesterday. So yeah, that's the thing. The main component of our molecular biology, this consists of basically three parts. That is your DNA that we have just discussed, RNA and protein. Yeah. So these are the three main parts which makes our main components, and together these three they make the central dogma of life. That from DNA, after the transcription, the RNA is produced, and from the after the RNA is being produced with the help of translation, your proteins are produced, and together this whole process is known as central dogma, central to the life. Yeah. So before I start to speak about gene and heredity, I want to take you some animations, genetic animations, which will be very lively and make our this session bit crispy. So what are DNA and genes? So let us see from this perspective of these ladies that we have just discussed just now. So I will remove my mic now. My voice will be not coming good as it is, but the voice of that lady.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order, but small differences in the sequence of the bases in our genes make each person unique.
So that's why me, you, and everyone is different. Our parents, we are different from our parents, but still we have 50% content from both of them. Um, just to make sure the animation that we have just discussed just now, it's give a brief animation of the same. Okay. So what are proteins then?
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
Konnichiwa. So that's the actually that's a Chinese art. It's not Japanese. They're doing in the morning these exercises. So it's just saying that after the functioning of DNA and mRNA, then many amino acids are produced, and together they have many functions in our body, and they work as they want. So a small video about protein. Cool. This one is very interesting thing. This always made me to think whenever I want to discuss about that how we can use this molecular biology in practical application. So this video explains everything, like we have the same gene but we can bring out a different function. For example, we can take the jellyfish, the gene from the jellyfish, which are fluorescent in nature, and we put them into the fish, a normal fish. so they will be glowing in the dark. The same, we can put them into the plants or into our cells in order to see the functioning of the organelles inside our cells. So it could be used the same way. The spider webs, they are producing these silk webs, so we can brought them into the yeast cells, and these yeast cells will produce in a huge amount of these silk web, and it could be used for various functions. So they are the same genes, but they have it's brought from the different organisms, and we can brought a really nice function for the human benefits. That's where your biotechnology comes. So we can benefit the human beings by the use of the nature, which is a good in a way.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing: animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
Very nicely and subtly explained about the same gene and different organism by our, I don't know who is this guy actually, I would love to know. Okay, so far so good. So all these things that I'm just sharing with you at the moment, so they are just bringing our foundation stronger so that we can uh, go further in more details. Okay. Okay, that's it. So I will bring to the next topic soon. So gene, as we know, it's a hereditary unit, as we have discussed in the videos, in which our whole segments of DNA, our RNA, all the material is present and packed inside the chromosome in this chromatin area. So in this colored area, so we have gene 1, gene 2 presence, which will be very beneficial for us. So our DNA with ATCGs, they got coiled together around the histones molecule, and after the coiling, they make these big balls. Also, again, coiling is done there. Then they got coiled together again into this very nicely packed into this chromatins, sister chromatids of chromosomes, and there our genetic material is present, and which could go further during the meiosis, transfer to the next generation. So I will not discuss this part about DNA here. We have discussed them already, so you can keep these lectures with you. Yeah, I want to discuss about RNA. So RNA, it's also a biological very important, in which there's not deoxyribose, there's a ribose nucleic acid. RNA was present, and it has a type of molecules that consist of long chain of nucleotides and units, and each nucleotide unit consists of nitrogenous base pairs, ribose, sugar, and phosphate group. Yeah. So this we can see it is a single chain amino acids of your RNA is going on. Then there's some double helix structure is happening, and they're making joint. Sorry, making a hydrogen bond here. Then again they become a single-handed. Yeah, single-stranded. Sorry. And then yeah, in this way your backbone is again sugar phosphate backbone like the all in the DNA, and you have a nitrogenous base pairs being attached there. So in this non-uniform structure the RNA has. There could be also double-stranded RNA, which is mostly present in your viruses. These double-stranded RNAs. So like in this, your protein code is outside in the HIV virus, and RNA as a genetic material is present in this double helix structure. So basically, from the uh, distribution point of view, there are three types of RNAs, that is messenger RNA, ribosomal RNA, and transfer RNA. So messenger RNA is used to code for proteins, whereas ribosomal RNA is used for translation as well as your transfer RNA, both together used for translation. And they are all present in most of the organisms, your messenger, mRNA, rRNA, and tRNA. Whereas for the post-transcriptional modification, in your post-transcriptional modification, the snRNA, the small nuclear RNA, it's used for splicing and other functions of your RNA, and it's present in the eukaryotes and archaea. And there's exceptionally yRNA and telomerase RNA, which are used for RNA processing and telomerase synthesis, respectively. And there's a regulatory RNA, also very uncommon one. There's antisense RNA, which is used for your translation block mRNA stabilization and used in the all organisms. So this is your messenger RNA works is it's uncoil your DNA. Then after uncoiling your DNA, it will start producing coding for the. There will be one coding strand. There will be non-coding strand. Your messenger RNA will start producing in the opposite of that, like for C, G, A. Instead of T, the messenger RNA will produce U here, U instead of T. So that's the main difference. So mRNA is then transported from nucleus to cytoplasm, where it is bound to the ribosomes. So after the translation is done, sorry, transcription is done, the mRNA from the nucleus, it's been transferred to the cytoplasm, where it is being translated again with the help of ribosomes and tRNA in order to produce your proteins. This step, single line, what we are saying here, it seems quite very nicely, actually beautifully written, but there is one complete video here. I will now start it now, which will explain the whole the region of your transcription, translation, and the central dogma of life, that is central to the unit of our life. So this statement will be explained now in this very nice animation.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
Now we will choose a gene, any gene.
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
So A will pair with which one for the mRNA? Students, tell me the answers. A will pair with for mRNA production. some saying T here. There's no option of T. There's no option of T because in mRNA T is not there. Its uracil is there. Yeah, it's U. Great. And then for the C, it will be A or G or U. For C, it will be these are the very basics, but sometimes when they comes in your exam, we tend to answer it wrong. So that's why I'm just making sure that everyone is on a good track, on a good way to go for ahead. So C pairs with G, and it goes like G pairs with C, and G pairs with C, T pairs with, yeah. If T is there, it will not pair with U. It will pair with the A. G with C, T with A with U, C with G, G with C, A with U, C with G, G, U, C, like this.
When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
So GGG, it will be UUG, CGG, CUC, glycine, glutamic acid, alanine, valine. So now what we are doing is your messenger RNA, it comes out of your nucleus and it's in the cytoplasm, and with the help of a small subunit and large subunit of your ribosome, and also with the help of tRNA, it started to produce your amino acids with the anticodons here. So if the mRNA is GGG, so what will be the anticodon on your tRNA? Will it be CUC, CCC, UUG, CGG? Which it will be, the 4 options? Glutamic acid, glycine, valine, or alanine? Tell me, students, what it will be. Yes, right. Very nice, very nice. It will be glycine. Now it's CUU. So it will be GAA. Right? GAA. Leucine. And it keep going on like this. CGG. CCC. So So here you can see the polypeptide chain of amino acids are being produced, and this keep going on.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, DNA helicase, will now slide between two complementary strands of DNA, temporarily separating them so that they can be copied. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
Very nicely shown, very nicely, adeptly shown this the transcription and translation, how it is done with the help of various molecules. So I will share this over the Telegram. So yeah, now we have just discussed this line in completely, that how your transcription and your translation with the help of your tRNAs, ribosomes, mRNA, DRNA, glucose, sorry, not glucose, your cytoplasm, nucleus, all these together, they're playing a very big role in order to understand our the basics of molecular biology, that these we are studying them for so many years in the books. Yeah, so even I was studying in my books, but no one was there to explain me with the animations or how they were happening in my body. So I was just writing them in my exam. I was getting good grades, around 80%, 90%. I was a bright student in my class, but in my inner Simran, there was always a fight, that how these molecules are doing their replication. How does it make why, how come during the DNA replication it's become a fork shape, so all these questions was just coming to me in my mind, and these animations they really help me out actually. So yeah, this is might be the things that are happening. So I'm just sharing the lectures from the student perspective. If I have been, because I have also done my BTech in biotechnology, did my masters in agrobiotechnology and then PhD. So I'm always thinking from the student perspective, that when I was student, whatever the things that I was feeling myself that I need to know. So I'm just bringing out, uh, outflowing towards all of you. Yeah, I hope it might be helpful for all of you. So okay, now comes the presentation part. So this is tRNA and this clover leaf shape, which is a small RNA chain of about 80 nucleotides, and transfer specific amino acids to growing polypeptide chains at the ribosomal site of protein synthesis during translation. So it has amino acid attachment site and anticodon region. So it has two regions that we have just discussed. And the site binds to a specific sequence on the messenger RNA chain through the hydrogen bonding. So here we have the codon region. This is the anticodon region on the top. On the top, the various amino acids are attached here with the help of ester bond, and yeah, this way. And this ribosomal RNA, it's of two types, 70S and 80S, which is of further is made up of 50 and 30 and 60 and 40 respectively. So this, I can skip difference between DNA and RNA now. That is the sugar base pairs difference, uracil and thymine main difference is there. There's a presence of hydroxyl group at the second carbon at the sugar group, and that is not present. RNA is mostly single stranded, where DNA is double stranded. Protein we have discussed in that video, which is a polypeptide chain of amino acids, which go and have a specific sequence of a gene and a code for specific genetic gene. So there are mainly 20 stranded amino acids are present. So together they make this polypeptide chain. So if it is starting from lysine, valine, phenylalanine, glycine, arginine, cysteine, glucine, glutamic acid, leucine, alanine, like this. And remember, between two cysteines, they are always making a disulfide bridges. That's what my PhD was. Actually, I was looking for the disulfide bridges between the two systems, whether they're present or not. So it took me 4.5 years, 4 years or so to find just these two. Yeah, I wanted the stupid disulfide bridges, but yeah, that was my hard work for my PhD, and uh, it just remind me my old days. Sorry, I need to come into reality. So yeah, in this way, these are the various proteins, and they go into the quaternary structure. You can see here two metal lines are present in the catalytic subunit with other various amino acids, and here is the beta helix state. This is alpha helix, alpha plated, beta pleated sheet, alpha helix sheets, and together they're making this quaternary structure. And these proteins are basically present in your peanuts, legumes, um, red kidney beans, fish, white chickpea, eggs, red meat, chicken breast. So just try to eat as much as proteins in your diet. Yeah, it's really beneficial. So basic players so far in our body that are playing role are DNA, RNA, and protein. Yeah, DNA, RNA, and protein. DNA could go self-replicate, and it could go to transcription to produce RNA, and from RNA your protein could produce with the help of translation. Yeah. So this we are discussing because sometimes in your interview somebody could ask, tell me the central dogma. Just don't say from DNA RNA is produced, from RNA protein is produced, and this process is no central dogma. So nobody explained that DNA could also self-replicate in order to do the replication, and the transcription could happen, and then translation is happening. And another way, there is a special central dogma also in which RNA could go back to DNA, yeah, with the help of reverse transcription, transcriptase enzyme, reverse transcriptase enzyme. And remember in this that from protein your DNA can never be produced. This is no. And also from DNA your protein could directly produce. This is also no, never. It never happens. And also from protein your RNA is produced. This is also never happens. So these three things are never happening within the central dogma of life. But yeah, in the exceptional cases, DNA could replicate itself, RNA could go back to the reverse transcription, and then from RNA, your protein could also have produced. Now comes the DNA replication. Okay, students, I think we could take off from here. How would you say? Would you like to take classes on the Zoom now for discussion, or can we discuss things here only? Do you want to take from here onwards to Zoom for 10 minutes in the last minutes? Because some students were asking for these Zoom classes, but unfortunately, I have the version that I have with me. Phew, I cannot have the Zoom classes. So here you can see it's not accessible for me. The Zoom is not accessible. You can't use this version of the applications because of the Mac OS. But if I um, make a new version of Mac, my whole this thing will be over. Hard disk is over. So I cannot use. I'm only available on the Google Meet now. So this DNA replication we can start from tomorrow. Yeah. So let's meet for some time over the Google Meet now. I will stop my class here. Can discuss here only. YouTube is good. Yeah, I mean it's good, but some of the students have been to the higher authorities, and they said we don't want classes to be on YouTube, and the standard of the classes are very poor. It's very basics. It's not of no use. We don't want our class. We want our money back or something like that. So some students were not happy with my classes initially. So it's okay. What we see sometimes it's not the reality. We try to we want to give you something from our own experience, but we want that the right time when we want to deliver the good things to you, we will deliver at that time. So I got some yeah, messages from some students that the classes were very basics. We don't want to study these things, but I deliberately put them thing because otherwise some students says students don't teach, teachers doesn't teach us the basics things. They just straightforwardly go to the advanced topics. We were not ready for that. So there are around 150 students here in the class, um, and it's very, if the class of is of like 20 and 30, there we can adjust with the things, but with the class of 150 plus, it's just a bit difficult, you know. Okay, there's a lot of chaos. Inertia's got disturbed of my house. Candy. Okay, then we keep the class here, but we should have sessions. I really want, we can have those sessions for our objective type questions then over the Google Meet. So once we are done with these topics of basics of molecular biology, I will have a small sessions. I have around 400 question banks of MCQs. So out of them I can share with you around 30 and 40 questions over the Google Meet, and we just discuss for half an hour so that we are good with that. Super. Thank you so much, everyone. Thank you so much. Thank you, Sangam. Thank you. Thank you, Harshita, Harshit. Hardly we will have tomorrow one more lecture, half lecture maybe. Then we are good to go. But okay, so we keep the classes here only. But as I started my class, okay. Anyhow, so we make this class here only. Then thank you so much. We see us tomorrow at the same time 8 p.m. And continue with the DNA replications. Yeah. Thank you very much. Good night. Take care. Bye bye.